Good day everyone, my name is Li Zixuan. So this video I will explain about the STPM Biology Chapter 2.1 Microscope Structure. So as you know that in the syllabus, the microscope that you need to learn is light microscope and also electron microscope. So these are, you need to know the principle and also their differences and also the type of the microscope that you use lah. Okay, so normally you see this picture. So the left hand side, this one is light microscope and this one is electron microscope. And so they are light microscope, they are bright field microscope or dark field microscope, and electron microscope containing two, scanning electron microscope and also transmission electron microscope, SEM or TEM lah. Okay, so you need to know their principle and theory and also their differences. Okay, so before you learn about the microscope, as you guys know that in microscope, the major principle is using the magnifying lens, right? Magnifying lens is the major component in within the microscope that we can see the thin structure, thin organism, which we call as microorganism lah. See the microorganism very clear compared to using the naked eyes. Our naked eye cannot see the bacteria, cannot see the cell, right? But microscope can. So microscope can. So there are two major terms that I think you also learn in physics in SPM. Right, resolution and also refractive index. So resolution is the measurement, the ability of the lens to distinguish fine detail and structure. Meaning that if the resolution is very high, meaning that the lens is able to let us to able to see more clear structure. Not necessarily magnifying. Magnifying is just enlarge the structure only, but not necessarily that structure is clear. Resolution can ensure that the lens give us can be able to distinguish fine detail so that we can see the clear structure. Not only just enlarge, if you just enlarge only, but the structure maybe is unclear. Like you take photo, right? When you just zoom it, yeah, it can enlarge, but the structure maybe is not clear. You need have a resolution. So high resolution, not only necessary it can magnify it, but it also can provide very clear and detailed structure so that we can see the cell in very clear one, not on very clear. And another term, refractive index, is due to the refraction process. So it's a light bending ability of a medium. So in general, your microscope should have the low refractive index rather than high lah. If very high, meaning that your light is bent away. Your light maybe can be go to another part, rather than go to the straight to the point. So this is the resolution. So in microscope, resolution is more high, more good. The higher value of resolution, the higher the fine detail, the clear structure we can see. If refractive index is, we need to as low as possible. Resolution as high, refractive index is as low. Okay, now we go to the principle of light microscope. Okay, so normally you can see this diagram. So this is the major structure, major component that when you arrange it. So as you still remember, light microscope is the microscope that normally you see it in your school. Your school use one, I think in SPM that you have use it. Not all of them is using light microscope. The electron microscope you cannot see it. You have never seen it before. Because electron microscope normally is used in the research one. It is very large and more complicated lah. Okay, so normally light microscope is just arrangement is like that. So normally you still remember that light microscope structure. You imagine it lah, like microscope structure. You imagine it. You place your specimen on the glass slide at here, right, at the middle part, and then once when you put it inside the sample, and then you were starting to rotate the lens, right, at the middle one, at the here middle one. So there are 3 lens, right? 10 times, 40 times, 10 times, 100 times, and 400 times. You still remember? Yeah. We will already forget. I think the experiment that you did I think it's the first experiment where differentiate between amoeba and domesticated or animal cell and plant cell. Later you see it. Okay, never mind. Okay, so you know that there are the gas light at here, and the major principle of light microscope is they are containing these two layers. These two layers play the major role in magnification, resolution, and visualization. If does not have these two layers, we cannot see it. This one, we cannot see the microorganism one. So, objective lens is responsible for both magnification and also resolution of the object. This one, meaning that this not only magnify, we enlarge the structure, enlarge that we can see it, and also provide the clear and fine structure, so that we can see the cell, the plant cell, we can see it clearly, or what is nucleus, where is the nucleus, where is the endoplasmic reticulum, where is the cytoplasm, very clear. If does not have resolution, like eyepiece lens, if only have eyepiece lens punya function, it only play the role in magnification only, does not resolve the image, meaning that when we see the object, the cell are very unclear. It is blur structure. So objective lens play the role is this, and eyepiece lens is play the role in magnification and also produce real image, the image that we can see through our eyes. Real image. Objective lens also play the role in real image, right? It just play the role to magnify it and resolve it. But eyepiece lens does not have the ability to resolve the image. It cannot play the function to identify, to distinguish the fine structure one. So if both of them use eyepiece lens, you cannot see the very clear structure one. You only can see the blur structure. Next is about the total magnification. So if you want to calculate the magnification process, magnification times, so this is a formula lah. You times objective lens, the number of objective lens, and also eyepiece lens. So normally eyepiece lens is 10 lah. 10 times, and objective lens there are 3, right? Either 10 times, 10 times, 100 times, and also 400 times. Yeah. So this one, I think in doing experiment, I think for one of the experiment that you need to calculate it lah. But I think your school teacher will help you. So this is just a formula that you guys need to know lah. And also, like this is the limitation of the light microscope explanation. So as you guys know that light microscope is only can see the microorganism, but it cannot to see the very small organism part, like bacteria. Bacteria you need to see lah, because bacteria is varying in size. Larger bacteria you can see it. So normally you can see that if the organism, the sample is less than 0.2 micrometer, so normally it's virus. For virus, you need to know that virus is very small. Under light microscope, you cannot see the virus one. You never see the virus. Only the electron microscope can see it. So why this limitation happen? Because of light microscope, I already mentioned light. So the major sources of this microscope is using the light. So the light is limited by the wavelength of the light. The wavelength of the light is normally 400 to 700. It is quite high now. Quite high. So due to the wavelength is very high, so that it cannot resolve detail one. Our lens, the lens is not able to resolve detail in the very finer structure, so resulting in low magnification. So compared to the electron microscope. And last one is the light also may bend into the air so much that miss the small high magnification lens. So emission oil is used to keep the light from bending. So as you know that you using the condenser lens, so normally your lens, your light maybe will bend out from the not enter to the objective lens, but your light is bent to the outside. So this is one of the limitations of the light microscope. So not all the light is focused into the objective lens. Some of the light maybe will expose to the other part, outside of the microscope. So normally that is why that some of the researchers, they will use the immersion oil to immerse it, so that the more light can be focused, can be passed through to the objective lens. Okay, so this is the basic principle of light microscope. So you see this diagram. So this diagram is that I just want to differentiate between dark field and also bright field. So normally in your school laboratory, school lab, you use normally I think is all of them is bright field lah. Meaning that your background is white. You can see this diagram. Your background is white color. But for the dark field, of course, your background is dark. So the principle is same. Just the difference is the under the condenser lens, they place the opaque disc at here. So that is why that you can provide the dark background rather than bright. So this is just a difference only lah. The difference only. Okay. And not only necessary for the light and dark field, there are also other type of microscope, but normally is similar basic principle of light microscope, just that they modify something. So you can see there are phase contrast microscope. The basic principle, they also containing eyepiece lens, objective lens, condenser lens. All are same, just that they are do some modification only. And you can see another one is differential interference contrast microscope, also same. You can see there are the objective lens, but this one is more focused on the computer lah. They use computer one. There are also objective lens, there are also condenser there are also eyepiece lens. So this one is you can provide more clear, and also you can see quite 2.5D structure lah. You can see there are 2.5D structure like this. So normally in the basic microscope that you use in your school normally is 2D. Of course 2D. But this one you can see like 2.5D like that lah. Okay. And another one, this one is fluorescence microscope. So this one is totally different, but the basic principle is still same. But they use fluorescence. So you can see they're still using the light, but the light is spatial, not the light that you use in your our hall. They use the spatial light, and also they use the fluorescence also. So you can see that they will provide this kind of structure. So more beautiful lah. You can see it's more beautiful, but still it is still 2D structure. More beautiful, they will provide more colorful structure. But they still 2D. So the major characteristic of light microscope is they cannot see the very small structure, but it can produce color structure based on the dye you use. You can use the dye so that you can do a coloring like that. This one is due to the fluorescence. Updating the sampling process, they use the fluorescence so that they can produce many beautiful color lah. Okay. So this is the black micro. Another one is confocal. This one is using laser. The light sources is using the laser. So it will produce the shape like that lah. Okay, so that's all about the light microscope. This is just an example that you don't need to know this all the diagram I just showed you only. The basic one is this. This you need to know. This one is you must know. This one is the basic principle of the light microscope that you need to know. This one is just an example I let you guys know only the extra information lah. Not only the light microscope not only have bright field and dark field, there are also many type of microscope that you use in the market now. Okay, now we go to electron microscope. So the view, you can see this image. View image of electron microscope is like that. So it is very large one. You know that your light microscope is just a small structure like that, right? Just like the computer only, here only, light microscope. But this one, electron microscope, is very large. So and then you need to use computer one. You need to use computer to set up everything. So it is very complicated to use compared to light microscope. So electron microscope, it is use the beam of electron to focus by electromagnet in a vacuum. So the sources of the electron microscope is using electron rather than light. That is the difference. Light microscope, the source is light, cahaya. The major source of electron microscope, of course, is electron. So they use electron. electron. So they use the electromagnetic magnetic that can produce the electron to replace the light. So the lens used is electromagnetic lens to focus the electron beam. So this electron, why you need to use electron beam? Electron beam can produce very short wavelength compared to light. Like I mentioned, due to the light wavelength is very high, 410, so it cannot produce very fine structure. We cannot see the fine structure. So, but electron beam have a very short wavelength, so it can produce very high resolution value. So normally it's at 0.5 lah, meaning very high lah. Very high resolution value compared to light microscope, so that it can see very fine and detailed structure even though virus. You can see virus. So all the virus sample like COVID-19, HIV, flu, influenza A, influenza B, H1N1, all that virus you need to examine under using this microscope. Light microscope, never see it. And this will cause high magnification and of the image is produced. And another character feature of the electron microscope is it only can produce black and white image. Light microscope, you can produce colorful image based on the dye used. But electron microscope, you only can produce black and white image only. You cannot see the colorful image under this microscope. Okay. As I mentioned, there are two types of electron microscope: DEM, transmission electron microscope, and also SEM, scanning electron microscope. So what are the difference? So the difference is the arrangement is quite different lah compared to here. You can see here. DEM normally they use the electromagnetic condenser lens at here, and also objective lens and also projector lens. So similar to this, they're also using the electromagnetic lens, but they use the amplifier SEM at here. Okay, so now we go to the transmission first. So transmission use more lens compared to SEM. You can see SEM just use two lens only. There isn't one type of lens, electromagnetic lens only. But you see DEM or use many lens. They not only use condenser lens, they also use projective lens, they also use projector lens. So they use three type of different lens, so that DEM produce more higher magnification power compared to SEM. So normally DEM can produce up to 10,000 up to 10,000 up to 100,000. Okay, 10,000 up to 100,000 times. So this one is totally different compared to light microscope. Light microscope, the maximum you can produce 4,000. 4,000 is already maximum. Because 400 times 10 4,000. The maximum 4,000. But you can see the lowest one, it can already up to 10,000. So DEM can produce very clear, that is the latest technology that use to examine the microorganism. So electromagnetic condenser lens normally is focused the electron beam on the smaller area of the specimen, and also specimen is placed on a copper mesh grid, and then so meaning that your specimen, your sample need to place on the copper mesh grid, right? Not like the light microscope you place on the glass slide. No. An electron beam will pass through the specimen and then through the electromagnetic objective lens, and finally go to the projector lens, onto a fluorescent screen or photographic plate. So the final image we call the transmission electron microscope. So normally it will appear as light and dark area depending on the number of electrons absorbed by different areas. So this is DEM. And another one is SEM. So also use the electron beam too, but the length used is quite less. So normally it's using electron gun. You can see electron gun. This one is using electron gun. So both of them using electron gun. And then they produce the electron beam, so that it will pass through the electromagnetic lens to produce the magnification process, and then it will directly go to the specimen. And then secondary electron beam are transmitted to an electron collector. electron. So image is produced on viewing screen or photographic plate. So the key difference is that DEM use more electromagnetic length. So that DEM can produce very clear and magnification power is very high. But SEM is only containing this one type of lens only, so that the magnification power is only 1000 to 10,000 only. Normally 1000 to 10,000. And the difference of the structure is using here. The SEM and also DEM. So the difference is SEM can produce 3D structure like this, very beautiful one. You can see it totally like 3D structure, solid structure like that. But DEM, sorry, it only can produce 2D structure. But DEM can produce very clear. You can see this cell, you can directly see there is the nucleus, and there are the ribosome, and there are the vacuole, very clear. But SEM, yeah, still you can very clear. But when you want to see the initial structure of the cell, it's quite difficult, quite difficult. But you can see very clear the whole cell structure. Cell, comparing to light microscope already lah. You can see lah. Okay. Okay, now this one is the differences between light and electron microscope that you need to know. So as I mentioned, your source for light is light wave. Wave, you will not only say light, you will just say light, no marks, light wave. An electron microscope is electron beam. So the wavelength used is normally light is 400 to 700. An electron is very low. You can say very low lah, as low as possible lah. 0.1 also can, 0.01 also can. And length used is light is using the glass, but electron is using electromagnetic lens, the spatial lens. And specimen used in light microscope normally it can be live specimen or non-live specimen. But electron one, when you want to examine the structure under electron microscope, you might use the dead specimen. An image, light microscope can produce colorful, but electron cannot produce colorful lah, black and white. And preparation, light microscope simple and easy, very simple. But electron is very lengthy and complex. And resolution very low for and magnification for very low for light microscope and very high for electron microscope. And cost, of course, if you want to operate light microscope, is very cheaper, very cheap, but expensive for electron. And the size is small and portable. You can bring it anywhere, but electron microscope you cannot bring it anywhere. You only can bring it at the lab. Okay, so that's all for this video. So hopefully you guys can understand about the microscope theory and structure. Thank you.